Friends, uh, good evening to all of you. Dr. Gandhi Garu, Chairman, Genome Foundation, Dr. Vaggu, Dr. Sharat Chandra, and other dermatology colleagues. The main objective of this Genetics in Dermatology webinar series is to provide a common platform for clinicians and geneticists to deliberate developments in DNA technology. At Genome Foundation, besides dermatology, we also have activities of this type of seminars in terms of infertility, gynecology disorders, and cancer. This is the third session of webinar series on dermatology. And when I said that development in DNA technology, the story is not yet complete. DNA technologies are emerging. Whatever cases we deal at Genome Foundation, it is not that we just give a genetic testing report. It is not the end of the story, because we are well aware that these technologies are developing and we need to be on a research footing to understand what are the developments in terms of, one is in rare diseases, genetics is playing an important role in terms of diagnosis. We narrow the cause of the mutation when it is known, and it can explain what are the path biology involved in this particular mutation. All the rare diseases, they are syndromic. Presentation is syndromic. Imagine a syndromic condition. We do have one single mutation, okay, which is being implicated as a cause of the disease. This is the advantage of rare diseases. And our experience in terms of rare diseases is that while finding the cause of the disease, there are other issues like family counseling. In collaboration with the clinicians, we do screen heterozygote status in the family members. For that, a small research fund from Genome Foundation with Dr. Gandhi's support, we do have this fund to screen the carriers. Once a mutation is known, to target the mutation and find in another family members, the cost comes down. From that angle, we are able to support few families which require some sort of support. And here, what is happening is that once carrier status is known, we are able to educate. All the processes in terms of collaboration with clinicians, and we could prevent the disease. That's what we believe. Family counseling and their constant touch. We are in touch with the patients, and also we are also looking into what are the developments that are happening in terms of therapeutic intervention. Okay? Some cases we do come across that once a mutation is known, total diagnostic scenario changed. Initially, it was thought to be ophthalmologic, xeroderma pigmentosa, but when the patient is a 9-year-old boy, when he went to ophthalmology LVPI, they suspected is xeroderma pigmentosa, but also they found on the skin some lesions. So it has come to Dr. Vaggu, and he referred the case to us, and we found a mutation, which is in fact a storage disorder, bile storage disorder. Then when we contacted a metabolic disorders expert in Bombay, and he was in fact telling to us, sir, this is, I am seeing this patient, which is only a 9-year-old boy. Okay. If you can give us folic acid as a supplementation, he will be saved from future, in terms of he was explaining all his patients are adults and who have been, already set in the neurological, whatever neurological problems, that is what the first expression. That's why this boy sight was the problem. Okay, but ultimately it come down to a metabolic disorder. Now he has been treated at folic acid. This is another story which is happening in the pharma field. Folic acid somehow production is not that common. Only in USA one company produces this, and it is costly. This patient is poor. Then when we searched and contacted different people, in Bangalore we came across one company which is doing biosimilar. And that person is able to supply the drug, and under the guidance of our metabolic disorder expert clinician, that boy is being treated. The boy's problem from the birth is he goes on for stools, uncontrollable motions. Okay, that's what his father explains. Sir, from the beginning, this boy has got this problem. Even while going to school also, in school also, he has to go repeatedly. Now, eyes was the problem. The teacher was able to detect, this boy is unable to read on the board. Then he referred to ophthalmology, and ophthalmologist, local ophthalmologist. This person is from rural Telangana, and they referred to LVPI. LVPI, there is a suspected diagnosis, and also they sent the case to dermatologist, Dr. Waggu. Now, Dr. Waggu, he has sent to us, he suspected something, wrong, because what I'm explaining this, because all these rare diseases are syndromic, which is just like a complex disorder. Okay, but because it is rare, rareness is because of the rare mutation. Okay, it is not that several mutations are causing the disease, but we are able to find out the causative mutation, which is driver mutation. It is not possible to get this type of driver mutations in chronic disorders, multifactorial disorders like cancer, diabetics, hypertension. Maybe at one time, in terms of human evolution, the cancer appeared. At that time, it could have been a major, driver mutation. Even in cancer now, there must be driver mutations. Okay, but the problem is several other small effect genes, we call it polymorphisms, they have set in, and driver mutation now for different phenotypes. Okay, it is also heterogeneous phenotype, and people are trying to find out these driver mutations even in complex diseases. That is what the recent research. But now we have rare diseases, whereas cancer, hypertension, diabetics are common diseases. Okay, because of environmental interaction, there are so many hypotheses, so many theory, so much of science behind. But rare diseases emerged recently because of technology improvement. So technologically, we are able to scan all the genes. Okay. Now, even though we don't know which is the causative gene, we are able to pinpoint the causative mutation, gene and mutation. And once we find the mutation, we are going back to the families to validate whether this is the normal person is having a heterozygote or no normal homozygote, whereas the patient is having homozygote and his parents are heterozygote. This type of, unless until we are continuously looking into what is happening in the field with respect to this particular disease, okay, and then intervention. Now we are happy at our Genome Foundation. And in terms of our collaborator also, when we exchange this information, they are also happy. The patient is benefited. Okay? And the future generation, imagine the boy, if it was not detected by us, by the early diagnosis, and if it continues for, ophthalmological xeroderma pigmentosa, I had a talk with the ophthalmologist. She said that she is planning for a cataract. Madam, please wait. This is what the findings we have. We are in touch with metabolic disorder expert, Dr. Jalan from Bombay. And, later we can think about planning cataract operations. Then now things are settling down. This boy is on this drug and he is responding. His father says, sir, he's going for stools has reduced. And this, ultimately the supplementation is going to be throughout life. Okay. But how much this costs? 7,000 for 100 capsules, the biosimilar. Okay. And whereas the USA, that original drug is very, very costly. So this type of intervention and going along with the patients is important when we are dealing with especially rare diseases where there is a possibility of finding the mutation, okay, cause of the mutation. We are successful in 98%, of all the cases referred to us. Certain cases, like Dr. Waggu remembers, that some cases, angioedema, there are, not mutation must be there, but our whole genome scan could not find it because whole genome scan has got its own limitations. Okay. What is required is we need to go for, in terms of whole genome investigation, where there is possibility of detecting large deletions and insertions. Okay. So there are some, cases which are challenging. Even that, in some, like mites case. Mites case is one thing that, the child when he's born, he goes on itching on his forehead. And that sensation is not there. He won't feel sensation. So forehead, you got a lot of itching and he goes on scratching. Ultimately, that will be wound and all that. And this is what, they call it mites and is a rare disease. And we went for genetic investigations. We could not find any causative gene. Only the gene, whatever is implicated, PDRM12 gene, has got 5 exons. We went for all 5 exons sequencing. We could not get the mutation. Then we looked into literature. Then I came across this Professor Woods, Jeff Woods, his group. They have published a causative mutation which has got large inversions on 5th exon. Then I contacted him and I was trying to ask him the details of the technology. Then he suggested, see, we are a research group. If you want to be a research program on mites, we can take it up. You can send a sample. Then I have called back Dr. Palasardi Garu, the ophthalmologist, and also Dr. Akhila Reddy, who is a resident, and she says that she was involved in first diagnosis of this case. Then with their agreement, we said that to my Dr. Woods, yes, Dr. Professor Woods, we can join your collaborations. Then he asked us to send the samples, and with all consent and all that. They are very systematic people. We are also systematic. We have consent forms, and all the subjects' consent has been taken, and clinicians have been informed. We forwarded it. And now, as the things stand, our clinician colleagues said that, sir, we will publish the clinical aspects first with genetics, whatever finding, with collaboration of Woods only. Let it be our publication. I said no problem. Woods, Professor Woods also agreed. Yes, you can go ahead with your publication. Okay. And who are the collaborators? He identified Dr. Pamela and Dr. Professor Woods will be there. So this publication is due. But what I would like to say here is that ultimately Woods lab, they found the same mutation what they found in other parts of the world. Okay. There are a few cases from West Bengal also. He was doing all over the world cases. And he is very excited to say, we found the same mutation, which is a large inversion. He has sent all the details to, I forwarded to Dr. Ms. Gargi to look into, and a publication has to come out with Akila Reddy and other our colleagues. So what is, my narrative is like this. Technologies are emerging. Okay, now we have this type of whole exome is very, very handy. It has costs have gone tumbled down. Okay, and earlier the cost will be very high. And whole genome, that's what next step. That is also the cost should come down another few years. And ultimately, as we could see, there is nothing beyond genes, DNA. Whatever is DNA is a blueprint of life. What you are, how you speak, how you sleep, what is your skin color, what is your hair color, all these things, have got a genetic basis. But here, rare diseases, it emerged only recently. About a few years back, we were listening to it because the genetics contribution to disease, in terms of other chronic diseases, not that much people are able to see in terms of diagnosis. Some cases, pharmacogenomics, the way the drugs are given, based on that, this polymorphism. We call them polymorphisms, These causative mutations are more than 100, so hundreds of mutations, hundreds of genes are involved, but they could find some importance in terms of drugs, even chemotherapy. Okay, such type of interventions are there. Now, again, in chronic disease also, things are coming down to finding out the driver mutation. According to path biology, drugs development, all this are going to be there. Besides that, they are also looking into what you call all omic approaches, integrated approaches. Suppose in the case which I described, whatever cases we have described, we are doing genomics, but there is something called proteomics. Proteomics is the protein, transcriptome. Okay, that is also people are doing whole proteomics, and then you have metabolomics. Okay, all metabolites, whatever DNA you have, whatever mutations you have, it should represent in some metabolites, chemical compounds. Okay, so integrating genomics and metabolites are going to give you some pathways or novel genes. Okay, how the genes work. This is one integrated approach is happening, and also microbiome. Microbiome, is another. We harbor millions, billions of other organisms in our body. Okay, and they also contribute. Imagine Parkinson disease. Okay, we know part1, part2, part3, several genes were identified. Now, what they found was that therapeutically, the microbiota, gut microbiota, okay, enriching some species is helping this neurological Parkinson disease. So such type of developments are there, such types of technologies are developing, and costs also tumbling down. So what one has to do, in whoever is doing genetic testing, whoever is the clinicians, they would like to have some interventions. What we need to do is some platform where this
Sir, good evening,
technology.
sir. Jeff Woods, sir, is online, sir, now.
Pardon
Professor
me?
Woods, Professor Woods is online.
Oh. Okay. I will conclude. I'm almost end of my story. So what is happening here? The platform, why we created all. In fact, Dr. Waggu was very instrumental in this platform. He said, we need to have monthly interventions, monthly webinars. I said, okay, then Dermatology Association joined. And it is a slow process, okay. But what I am, very much positive about this is, among the clinicians, we have people, okay, who are into like understanding genetics. They all know genetics. But technology, technology is not one man's, only one man is only responsible for technologies. Technologies are available, okay. Abroad, I think Professor Woods is a pediatrician, he also into human genetics. And they have a very Cambridge, very good research group, okay. Rare diseases, Mendelian diseases. But India, whatever you think of, it is only in government setup, okay. That is a different story. I don't want to go prolong on this. With this, there is a lot of scope and lot of education, awareness. Pardon me. So this is what I am. I will now conclude. Professor Jeff Woods has joined us. I'm glad that he's around, and I'm thankful to him for accepting our invitation to participate in this webinar series. And now I thank Dr. Gandhi Garu, our chairperson, for facilitating all these processes, And if Dr. Gandhi Garu want to share some of his thoughts, now Dr. Gandhi Garu, you can come over and share few thoughts. Then Dr. Waggu will introduce the speaker and welcome him, and our professor will deliver his talk. Thank you. Dr. Gandhi Garu is available? Hello?
Good evening, sir. Good evening, sir. Dr. Rao, sir. Dr. Gandhi just stepped out thinking that respected
Doesn't
sir
matter.
going to join us. But I told him to join back in like 2 minutes.
Okay, shall I wait?
He will be joining in 2 minutes, sir. He just stepped out thinking.
Yeah. Meanwhile, let us have some sort of chat with our professor. He's online. Professor?
Hi, I'm here.
Hi, hello. Are you on video or?
Yeah.
Yeah. Garge, can you focus, Professor Woods? Yes, Professor Woods, good evening. This is evening here in India. I'm glad you are you are there online. We are surprised where you got missed. And thank you for coming on a Sunday. And what is the time there now?
It's 2:00 in the afternoon.
Afternoon must be it's not a working day for you. Even then you accepted. That is great. As I was explaining, we are trying to get into in terms of giving a platform for clinicians and or geneticists, okay, to have a dialogue in terms of development of technology. The DNA technology is developing, it goes on. Still, there are so many issues there. Rare diseases is immediate interventions possible, and we need to look into this type of educating and awareness about clinicians. And patients also need to be, in terms of family counseling. We are successful in several very interesting cases. I was narrating to them all few cases. Now I'm glad you have come back. And our chairman, Dr. Gandhi,
He's online, sir.
who is instrumental, okay? He is would like to share a few thoughts. After that, Dr. Waggu, our dermatology consultant, he will introduce you and you can take over. Thank you, Professor. Dr. Gandhi Garu, please.
Yes. Good morning or
Good
your
afternoon.
time. I think good afternoon to you, Dr. Jeff Woods. And thank you for joining us. We have been eagerly waiting to listen to you. Our Dean of Research, Professor Rao, was in touch with you, and we have been anticipating to receive you and then listen to you almost for last 5-6 weeks after he contacted you. Without coming between you and the listeners, I thank you for taking time and joining us, and we'd like to continue our association with you, especially because in this area of dermatogenetics, we are trying to make some focused studies, and already we have been successful in identifying more than 20 rare diseases, and our experiments are continuing. There are a few dermatologists, including the association, Indian Association of Dermatology, who have been supporting our work. And today, although not many people have joined because of, I think, Krishna, there is some festival, Krishna festival in India. So I'm sure that whatever you speak will be conveyed to the rest of the people, and it's being recorded. We'll try to make it make the abstract available to other people also. And I request that your association will continue for Genome Foundation to enrich the knowledge and contribute to society as a whole, humanity as a whole. Thank you very much once again. I welcome you to Genome Foundation's webinar today.
Thank you, Dr. Gandhi. Dr. Waggu, Anand Kumar ji, please. Yeah.
Good evening, all, and I think good afternoon to Professor Jeff Woods. And yeah, this Genome Foundation, along with Genome Foundation, this is a third seminar, third web series, you can say. And first two was done very successfully. I'm a practicing dermatologist, a clinical practitioner. I used to refer a lot of cases of very rare cases to the Genome Foundation. With help of them, we could able to diagnose a lot of difficult cases, so that especially I can say is a moldy melida, and one recently Dr. Rao shared about the suspected geroderma pigmentosum, but it is turned out to be something else, and hereditary angioedemas, lamellar ichthyosis, so that it is not only important in diagnosing the disease, so it is very important genetic counseling to the parents also, so that they can understand the disease process and they can plan their family in future also. So definitely, especially in moldy melena syndrome, when he diagnosed it as a moldy melena, because previously he was used to treat it as psoriasis with methotrexate, then we diagnosed it as a moldy melena. Then we stopped methotrexate, and that time her wife is pregnant. So that then we counseled, we did genetical study, and antenatal diagnosis of moldy melena was also done. So that will definitely will help in the patients. Not only for the disease processing, genetic counseling also it will help. And but today, Dr. Jeff Woods, I would like to invite Dr. Jeff Woods. Dr. Jeff Woods is a professor. He is trained as a pediatrician, then a clinical genetics in Birmingham, London, Oxford, and then spent 2 years at the Murdoch Institute in Melbourne, Australia. A missing home, he returned to the UK to work in Leeds for 10 years, predominantly with the consanguineous population of Yorkshire. He built up a research team with autozygosity mapping expertise and concentrated on families with primary microcephaly. Reluctantly, he moved to Cambridge in 2005 and moved the focus of his research to the genetics of pain and somatosensory disorders. So I would like to invite Dr. Professor Jeff Woods to talk on with extreme phenotypes, think of genetics, even with itch.
Thank you very much. I'll share my screen if I may.
Yes.
Can everyone see my screen?
Yes.
Yes.
Right. So firstly, thank you very much indeed for inviting me and making the time. I have not been interested in itch until about 10 years ago. And so I am very naive about itch compared to you who are very experienced about itch. So forgive me if I make mistakes. And I'm going to talk about clinical genetics and genetics, but also about three what I would call extreme phenotypes. I've spent half my time as a clinician and half time as a researcher, and I have my research team here at the Cambridge Institute of Medical Research, and that's within the University of Cambridge in the UK. And as a clinician, it's an excellent place to work because there are so many people that you can collaborate with, because no problem in medicine or biology is simple. You need friends. So just a bit more of my background. So as I said, half my time I'm a clinical geneticist, and so I'm interested in the inheritance patterns of disease, and I'll show you a dominant and two recessive diseases today. I'm very interested in phenotypes. Some phenotypes are very difficult to define, such as the psychiatric diseases like schizophrenia. Some are easy to define, like children with severe learning difficulties, but we all know there's hundreds, if not thousands, of causes. And some phenotypes, like the ones I show you today, I think are so specific, you can say that the phenotype alone defines the disease. And finally, I'm interested in genotypes. I'm interested in what are the genes and what are the mutations in those genes which cause phenotypes in humans. I don't work with mice or other animals, but I have colleagues I collaborate with who do. And I did not start off as a researcher to look at pain, but I sort of got pushed towards pain because of patients I saw in my clinic. I saw a number of patients in Bradford in North Yorkshire when I was working there who had no pain from birth, had congenital insensitivity to pain. And so we've studied this condition and the genetic causes of it. We also look at some of the types of chronic, and again, we're just after what are the genetic factors which make some people have chronic pain and some people are resistant to developing chronic pain. And we also have an interest in visceral pain. The commonest severe pain, as you'll all be aware, is labor pain, which is a visceral pain. There are many others. And visceral pain operates completely differently to most pain, so that's why we're interested in it. But now we're more onto our talk. When I was preparing for this talk and thinking about it, I realized that in English, the word itch both means the sensation of an itch, but it also means what you do in response to an itch. You itch yourself because of the itch you have. And I have no idea how many words there are for itch in the Indian languages, but it's really odd. We just have one word. And we also have, you can say, I've got an itch to find something out when you have a desire to find something as well, sort of showing that itch is a strange sense that it drives you to do something, which is to scratch. And we were always amazed when we started to work on pain that pain wasn't regarded as a sense. Of course it's a sense. It's something that's detected by the nervous system. There's whole systems of cells in the nervous system do it. And itch is the same. Itch isn't regarded as a sense, but it is. There is an even bigger neurological system in humans for detecting itch than there is for pain. And it's amazed me that itch has not been taken much more seriously, because it's a very, very common phenotype. And the other thing that's odd about itch is, again, if I compare it to pain, which I'm more familiar with, there are different types of pain and they can have different severities, but itch seems to be only one feeling. All stimuli for itch just produce one sensation, and all that varies is its severity. So it's a strange sensation. It's not like vision, which we know is very complex. Itch in some ways has a single readout, which is the desire to scratch yourself. So on those few thoughts, which I'm sure you've all realized because you're dermatologists and think about it, I'll move on a bit. So if I was talking in the UK to dermatologists, I would be apologizing for talking about one of their conditions to them. And in the UK, these would be the type of things that you'd be thinking about if someone came to you with itch. They're obvious: allergies, atopies, skin disorders like eczema and psoriasis, insect bites, huge cause of itch in the UK. Renal failure, most people with renal failure will develop itch. Although we find in liver disease, especially some diseases like sclerosing cholangitis, the itch can be the most dominant feature and the dominating feature of the condition. And obviously fungal infections. And you can possibly not see down here, right in the very, very smallest letters, genetic causes of itch. And these are indeed rare, but it's what I'm going to talk about today. So I'm going to tell you 3 stories about itch. And this is a picture I usually use in pain talks for the pain and the despair in chronic pain. But I think the people I'll tell you about today would probably look fairly similar if you saw them and saw that, which I'm sure some of you have seen them and seen how severe itch can be. And I'm going to talk about a family with dominant itch, a very rare condition called SCN11A itch, and something that I know that you're very good at diagnosing in India, Meigs midfacial toddler excoriation syndrome. So I'm going to start off with talking about a family who was recently referred to me with congenital itch. And this family have autosomal dominant facial sparing congenital itch. And I'll show you a pedigree to show the autosomal dominant. The facial sparing is, as you'll see later, very interesting. Why these people have itch all over their body except for their face is puzzling. And congenital itch, that is, they were born itching and they never stop itching. So this family was initially diagnosed by Nigel Burrows, who's a wonderful dermatologist at Addenbrooke's where I work. And he's very experienced. And when he was sent a family with several people with itch, I think he was very clever to distinguish initially that this wasn't just a couple of children with idiopathic itch or eczema itch. It actually was a potential genetic disorder. And initially he saw a mother and her 3 children. And 2 of the sons had something I'd never heard of called nodular prurigo, which I guess you guys are very familiar with. And straight away, Nigel realized that there was something very strange here, because nodular prurigo is supposed to, certainly in our population, to be an adult disease, particularly younger adults. And the majority of them, you can be fairly sure, have atopy. And whatever you do, it's very difficult to treat. And there's been quite a bit of debate in the literature about, does the nodular prurigo itch, or when you scratch yourself enough, do you develop nodular prurigo? And I guess our family would sort of suggest that actually nodular prurigo is a particular reaction of some people to itch. Let me show you the family. So this is a classic dominant family. And I know you're probably all very literate in genetics, but just to be sure, dominant family, which are the affected people filled in as females or males. You can see it passing down from females to females, females to males, and males can pass it to males, or males can pass it to females. So in this family, and there is in fact a third branch, which I haven't drawn here because we haven't seen them yet, all these people filled in have the same phenotype. So they have the following features. They have itch from birth, and I guess immediately you'll say, well, how do you know a baby's itching? Which would be a very, very good question. And that's exactly what I asked the people I've seen. And what happens is the family can tell who's got the condition or not, because as a baby, the baby continually squirms. It rubs its legs together, it rubs its arms against its body, and it squirms in the cot, which has always been assumed to be trying to quell the itch sensation. The males are as severely affected as the females, and although I showed you two boys who both have nodular prurigo, further in the family there are females with nodular prurigo as well. The itch is severe and dominates their life, and it is utterly unrelenting. There's reasons, as I said before, I'm really not sure about. It spares the face, and no one seems to have significant itch of their face. It's everywhere else. They itch in their sleep, and so if you're married to someone with this condition, they will keep you awake because they are itching themselves all the time. And we also have one very sad case. It was this lady over here. Can you see my pointer? I think you're muted, Professor Rao. Can everyone see the pointer?
Yes,
Oh, great. This lady was dying of cancer, and she was in a nursing home, and the family were there seeing her. And they gave her opioids, and she gradually lost consciousness. But everyone had noted in this place that during her sleep she itched. And when she became unconscious with the opiates, she just continued itching. So this itch is very unusual. It's breaking through something like sleep, which switches off many neurological diseases. The family all say heat makes things worse. So this family, they would want to wear sandals rather than shoes. They'd want to never wear hats. They'd want to wear very light clothing, and they love the winter. They hate the summer. And no treatment. Everyone in this family has had multiple treatments, and everyone said you got eczema or you got something else. And they've had lots of steroids and treatments for scabies and treatments for fungal infections, and nothing has made had any good effect. Steroids particularly don't have any effect. But what they hate the most is ointments, when they're given ointments and creams. Does anyone know why they hate ointments and creams? This is not a trick question. It's a real question.
It becomes worse.
No, they don't become worse.
Okay.
What would you and I do if we itched a lot?
We put ointment, or we wash it.
Think a bit more primitively. What's the instinct if you itch?
Scratch.
You rub it to a hot surface.
Sorry, someone said something else?
Okay.
They hate ointments and creams because they cannot scratch themselves hard enough to get pain to relieve their itch. And so if they have ointments, they can't, if they can't bear it anymore, they can't scratch enough to cause the pain. And this family is interesting because they feel pain normally otherwise, but they use pain if they can't bear the itch anymore. They use sharp knives to stroke up and down their arms. One of this young girl here, she cuts bits of skin off the soles of her feet to relieve the pain of her itch. The nodular prurigo is undoubtedly itch related. And as they get older, they learn the only way to help themselves is to mentally not scratch. They know the more they scratch, the more they want to scratch. And so a lot of the family members actually avoid medics because they know nothing works, and they don't want to be given creams and lotions. They want to be able to occasionally scratch themselves. So this is an incredible family. And I think what was for me very gratifying, but also very good, was everyone I talked to told me the same story. And there's, as I said, there's 3 branches of the family. And as you can see, they're actually quite distant because the intervening relatives have all died. The 3rd branch of the family, in fact, the 3 intervening relatives have died. And so they're not all talking to each other, but they all have this incredible history of severe, unrelenting, continuous itch. So there are actually 2 published dominant forms of itch in the world literature, which surprised me. But one was by someone called Steve Waxman, who's very famous in the pain world. And he works on a pain gene, one of which is called SCN10A. And some people who have SCN10A mutations have paroxysmal erythromalgia. So they have times where they get pain, and then where they had the pain, which is usually legs, they then have flushing erythromalgia. And that's quite an unpleasant disease because the episodes of pain get worse and worse as they get older. And about 1/3 of the people with this condition will commit suicide because it's untreatable. But there is one family who had a particular set of mutations where they had paroxysmal burning followed by itching, which was said by the family to actually be worse than the burning. So that's one very rare cause. But this type of itch develops when they're in their early 20s. They do not have the disease when they're born or do not have symptoms of the disease. And then there's a second report, which I'm not quite so clear about, from Italy, 3 very small autosomal dominant families where children developed this condition about the age of 7, where they got itching, but it was only on their trunks and their arms. And in this case, gabapentin, which is very useful for almost all neuropathy conditions, did cause some reduction in itching. So there was some history in the past that there's been some dominant families, but nothing like our family with congenital itch. And so we did what's called an exome, which I guess you're all very familiar with. It's a test on your genomic DNA, and the aim of an exome is to sequence all the exons in genes, because that's where the vast majority of mutations are that cause Mendelian disease. And so we used an exome, firstly, to rule out the 2 previously known causes of dominant itch, COL6A5 and SCN10A, and then secondly, to see if we could find the cause of the itch in this family. And we haven't been able to yet, but we do know they have over 2,000 previously unreported mutations in their genome. So we're going to slowly work through those to see if we can find out what in the world is going on with this family. So I never thought I'd ever come across a family like this. And I just wonder how many more families there might be out there which just have not been diagnosed, because itch can be such a common condition, but it's this extraordinary distribution of the itch and the fact it carries on during their sleep, and there's the dominant family history. So if you have any families, then I think you've got an interesting and important family, and you should work on it. So that's my first family, which sort of very much surprised my colleague Nigel Burrows when he came across them. He sort of didn't believe it could be a dominant family till he just started going through the pedigree carefully. Okay, I'm briefly going to talk a tiny bit about pain and itch now. This is from a website called Mousebrain, and what it does is they've sequenced all the genes that are expressed in messenger RNA in all the cells in the central nervous system, which are mostly at this end, and the peripheral nervous system, which are mostly at this end. And here are where the sensory neurons are. And as I said, I study pain. And so PRDM12, this gene here, causes painlessness when you knock it out. And so it fits in this area here of the pain neurons. SCN9A, again, if you knock it out, you're painless. Oh, excuse me. SCN10A, forget that. SCN11A, I'll show you mutation in that later on, which causes painlessness, and that's sort of expressed. But as you're probably noticing, all these genes that cause painlessness are also highly expressed here. And these neurons in orange here are the itch sensors, the Puri receptors of the peripheral nervous system. And down here are a couple of genes which are very specific to different types of itch receptors, proreceptors. And so you can see actually most of my pain genes are actually very well expressed in the pain receptors of the human body, the nociceptors, and the proreceptors. And it's very interesting that pain is mostly peripherally received by neurons in the peripheral nervous system and then sent up the spinal cord. Itch seems more complex. Itch is perceived by a whole number of different types of receptor cells past the spinal cord and then is processed in the spinal cord before being sent up to the brain. And it's maybe this reason that if you scratch yourself to cause pain, it can somehow gate out the itch sensation. But there is a very big molecular relationship between pain and itch. So I'll move on to my second type of extreme phenotype, extreme itch. In some ways, I apologize for this case because I think there's only 4 people in the world with this condition. And this is a particular mutation in a gene called SCN11A. SCN11A is a voltage-gated sodium channel, and it's the 11th of those genes. And the mutation is heterozygous, that is, these people have one normal SCN11A gene and one with a mutation, and it's a very peculiar mutation. Leucine at position 811 is changed to proline. This mutation is always de novo dominant. That is, both parents don't have the mutation, and in all the reported cases, both parents don't have the disease. The child is the first person in the family to have the disease and has the de novo mutation. And this mutation is thought to be a hyperactivating mutation. I'll talk about that in a second. So this child, you can see, doesn't look normal, but in fact, they are normal in appearance, but what they do is have significant dystonia. You can see the strange posture the child's got with their legs, and that child is grimacing. And as they sit there or stand there in front of you, their face is always contorting and grimacing. So one feature of this condition is grimacing. Here is the protein. In fact, I'll come back to that in a second. So this is a long list of the features, but these children present with being unable to pass motion, and because of that, they become chronically constipated, and because of that, they start to vomit, and because of that, they can't feed. And these children spend their first year of their life in hospital normally, because they either have to be parenterally fed, IV, or they have to have colostomies or gastrostomies put in and be very, very carefully and slowly fed. And when people have looked at their bowels are not peristalsing. They're just sitting there doing nothing. And gradually over the first year, they start to get some peristalsis, and during years 2, 3, and 4, their bowels start to work. They are always constipated throughout life, but in the first year, undoubtedly in the past, some have died of chronic constipation, but they nearly all need parenteral feeding. The second feature is it becomes obvious they're painless. They never feel pain at all. The third feature is itch, and I'll show you that in a second. They get itch, which is always present, and it's very temperature related. They hate higher temperatures, they hate lower temperatures, but there's probably a 10-degree area, 10 degrees centigrade area, where their itch is at its least. Make them hotter and sometimes make them colder, the itch gets more severe. And the itch is very peculiar. It's on the back of the neck. They feel itch all over, but the worst place for them and where they always itch is the back of the neck. And this is not the picture I would love to have shown you. This is the girl in the first slide. And this is the back of her neck. When it had healed, she had scratched down to the muscles of the back of her neck. So bad was the itch. And then her hands were tied. She was put in mitts. They covered the area, they let it regrow, and then they had to do some plastic surgery. And so her itch was so severe, she scratched through to muscle. The intelligence of these people is normal. And what is quite extraordinary is that colleagues in Germany introduced this mutation in SCN11A into mice. And here's the mouse. It's again scratches itself, and it scratches itself on its neck. It's quite an extraordinary phenotype. Itch all over, which is sort of containable, but for some reason, itch at the back of the neck is excruciatingly severe, and they cannot help but destroy their own tissues. Just popping back to this, just to explain, if you think of this as the outside of a cell, this is the cell membrane, here's inside a cell, and this is an SCN11A molecule sitting in the membrane. And what it can do is let sodium pass from outside the cell into the cell. And this part of the molecule makes sure that it's only sodium and not calcium, not potassium, not chloride, not water molecules, only sodium molecules that can flow through. And this part of the molecule can open and close. If you think of this molecule like a donut, where the donut can squeeze itself shut. And the amino acid right at the bottom here is this leucine, this leucine here, and this is the leucine that's mutated in this condition. And the thing about proline is proline is a rigid amino acid which kinks the peptide chain of a protein. And so if you have a proline in a position, it will always hold your protein, for instance, like that. It won't allow it to be flexible at all. It'll always hold it in one position. And what happens with this mutation is it holds this part of the protein open. And so in the neurons, SCN11A is allowing a constant flow of sodium into the cell. And so neurons, which are nociceptors particularly, can never fire an action potential because they're always depolarized. So this is a single mutation disease. It causes lifelong severe painlessness, lifelong severe itch, but for, again, no reason I can explain. This affects the back of the neck only as the most severely affected part of the body. And the third condition I'd like to tell you about is mites. And I got into mites because of this child and their condition. This is a child with congenital insensitivity to pain, and you can see that she's bitten off bits of her lips, and in fact she's bitten off the front third of her tongue painlessly. These are two children, brothers, I think a brother and a sister, sorry, with mites. And what you can see here is the end result of the mites, which is, my apologies, that there is now discoloration and hyperpigmentation from where they had their injuries. Now, the reason I'm showing you these two conditions will become clear. So I'm going to go back to pain for a second. I talked about that child who had congenital insensitivity to pain, and she had it from mutations in this gene called PRDM12. It's a transcription factor. And this is someone with the condition. He's bitten off that bit of his tongue. You can see he's lost fingertips from this condition, thumb tip. You see some tongues which have been mutilated here. They get a neuropathic keratitis of their corneas, and so they do not know they're getting corneal damage, and they can get severely scarred corneas. If those corneas are transplanted, the transplant also doesn't sense pain and again gets damaged and passes away. They mutilate their lips, they have problems with their fingers and toes, and a lot of the children with congenital insensitivity to pain are very sensitive to Staph aureus infections, and mostly these children have had trivial injuries to their fingers or toes which have become infected with Staph aureus. The Staph aureus has then got into the bones and caused osteomyelitis. So this is a form of congenital insensitivity to pain where you do not feel pain, you have increased Staph aureus infections, your intellect is normal. So here is the PRDM12 gene, and various mutations shown here can cause this condition. You have to have a mutation in both of your genes, as this is recessive. And this is the family which we first ascertained with this condition. This is a Pakistani family. They all live in Bradford. You can see there's a number of affected individuals, and they're all related to each other. However, we actually missed the mutation that caused the gene in this family. We looked at this family for 5 years before finding the gene. This single family here, which actually is this gentleman here, we found 2 mutations in him in PRDM12. We then went back to this family because we knew the PRDM12 gene was in the region we were looking for the gene, and discovered what they had was an expansion of a polyalanine tract in the protein. So a polyalanine tract would be a string of amino acids alanine repeated, and you can tolerate up to about 13 alanines in a row before it causes problems. But after 13 alanines, it almost always causes protein aggregation and a loss of the protein. And in this family, they were homozygous for 19 alanines being present. So I want you to remember 19 alanines in this PRDM12 gene. And that causes you painlessness and nothing else. So about 5 years ago, this is Celia Moss, who is a professor of pediatric dermatology. She's now retired, but is still active, from Birmingham. And she, with help of Indian colleagues for the most, reported what she thought was a new condition, mid-face toddler excoriation syndrome. And there was a lot of skepticism when she presented this in meetings to begin with. So what it appeared to be was children who itched a lot, but didn't have itch elsewhere. So they had excruciating itch from birth. Strangely, the itch seemed to be at the ends of the trigeminal, ganglion, mandibular, and ophthalmic sensory branches. And I'll show you some more pictures later. Itch elsewhere in the body was completely normal. So it wasn't like the normal itchy baby you see in clinic. The itch was nowhere but the face. And the itch, although they scratched their faces, they didn't seem to get Staph aureus infections. All other senses, including pain, were normal. And as we've studied more of these children, it's become very obvious that the itch starts to decrease during the first 10 years of life. It becomes more bearable. And by 10, they're often able to control it by willpower alone and the want to not look deformed. And the scars then heal, and heal quite well by the mid-20s. In fact, we've got a couple of cases in their 20s, and one of them you would never have thought he had anything wrong with his face. And this is an autosomal recessive disease. So some pictures. So this is for me, a very, very instructive picture, brother and sister. So you can see this X-shaped distribution of where the itch is. It runs down either side of the nose, up onto the forehead, and occasionally on the bridge of the nose. And this is the distribution of the itch, and this is where they scratch. So this little boy has been scratching, and you can see some quite bad damage here. The sister is managing to stop herself scratching most of the time, and she's got the hyperpigmentation stage, but her skin is now starting to heal. And when you talk to people like this girl, she'll tell you what it was like when she was a child. She was never without a terrible desire to itch and scratch her face. And the only thing that could help it was the scratching to cause pain. Here's a lot of pictures that were in one of the first papers about the condition. So each time you're seeing pretty much always the scratching on this X sort of shape, sometimes in just one smaller area of it, sometimes more extensively, sometimes the bridge of the nose as well, but usually not. And here's a nice set of pictures. For me, it's nice anyway. 14 months at presentation. wasn't diagnosed at that time. At 4 years, the itching was still happening, and you can see it's much more distributed now. And this is when we made the diagnosis. And here is at 6, where the family and he are trying very hard not to itch. And so he's been left with the hyperpigmentation, but he's not got any more injuries, which are often penetrating. And we have one child who has scratched through to his turbinate bones in his nose with this condition. Here's another photo. We've been trying to do some follow-up studies on our children, and from when they were diagnosed to older. So this little boy was diagnosed, I think, when he was 2. And this is a very typical facial appearance. And notice how the cheeks are spared completely. Under the mouth is spared. Most of the forehead's spared. And yet what he's been left with is this terrible scarring and hyperpigmentation, which for him is an awful blight. And he's changed schools to avoid bullying. And he has now a huge drive not to scratch himself because he knows the scratching causes the problem. And he uses cold water an awful lot of the time to try and help relieve the itch. And also he actually squeezes his own fingers to cause him pain in his hands to avoid him trying to scratch himself. So a quite incredible condition where you have itch from birth driving self-mutilation, but in a very defined distribution. So you were holding on to the fact that congenital insensitivity to pain can be caused by having mutations in PRDM12, the gene PRDM12, with 18 alanines. And all those children I've shown you with mites have homozygous 19 alanines in sensitive to pain, 18 alanines in mites. So 18 alanines gives you mites where you scratch your face and itch your face terribly, but not the rest of your body, and you have no problem with pain. Your pain sensing is completely normal. And then one more alanine, 19 alanines, and you have completely normal itch, and you itch normally with insect bites, etc., and you have complete loss of pain. And this is again an extraordinary example of a single mutation disease in mites. Only this 18 or 17 alanine repeat will give you mites. 19 alanines is one of many mutations which will give you congenital insensitivity to pain. And it's very difficult to work out why such a small difference in a mutation would cause such a massive difference in phenotype. This is just a picture of normal cells. The blue shows the nucleuses, and in red shows PRDM12. And this is normal PRDM12 with, I think it was 12 alanines, and it's in the nucleus as it should be. And this is one of the families, either with the 18 or the 19 alanine repeats, doesn't matter which, they both give you the same result, that rather than this fuzzy pattern, you get the aggregation of the PRDM12 protein. And in some cases, the protein, the aggregates escape the nucleus and in the cytoplasm. So mites and congenital insensitivity to pain due to PRDM12 are both caused by protein aggregation. But as far as we can tell, there's no difference in the amount of protein aggregation, but one gives you the itch, one gives you the loss of pain. This is a diagram made by Nivedita Sazatula, who actually did most of this work. And it's really pointing out that if you look at the trigeminal distribution, the trigeminal ganglia, which is in fact a very complex ganglia, it's a motor and sensory ganglia. Most sensory ganglia are only sensory ganglia. The itch is sort of mostly at the extremes of each of the trigeminal regions, between ophthalmic and maxillary and mandibular. Sorry about that. So that's mites for you. We have no idea why 18 alanine repeat causes such a localized extreme itch just in the face. Just like we have no idea why SN11A mostly gives you itch at the back of the neck, and the first family I showed you gives you itch everywhere apart from the face. But these are extreme forms of itch, and I guess I call them extreme because they're self-mutilatingly severe forms of itch. So if I have any messages to you at all, it's there are still new diseases to discover. Don't give up trying to find new things. And if you see something strange, treasure exceptions, because you might have found something new or recognize something which is old, but recognize it for the first time. I don't need to tell you as this audience, but if you see anything which is abnormally severe as a disease, think about it being a genetic form of that disease. And finally, I think we've increasingly realizing that sometimes a single mutation can cause a phenotype, and I've shown you two very good examples of that today. I guess, have I said anything which you think is wrong or doesn't chime with what you already know, or am I preaching to the converted when I say that itch is not a benign sensation?
Thank you, Professor Jeff Woods. Very interesting story. And let us have some comments from the participants. Friends, do you have any comments on Professor Jeff Woods' lecture, whatever he talked about?
Professor Woods, if you don't mind me asking, when you're saying that the pain is better as a suggestion for those families for scratching or scratching the itch, are you talking at any part of the body, or is it specific to a region, or is it just any type of pain?
So you're absolutely right to ask that. So they are using itch to cure, sorry, they're using pain to cure the itch, because everyone, I think, has the sensation that if you have an itch, if you scratch it hard enough, you get rid of the itch, but actually you're causing pain, which is somehow a dominant sensation. And so these people, we've always assumed they itch and scratch the part of their body with the maximum amount of itch. Sorry, there's a problem with the word itch having two meanings, isn't there? So yes, they're using targeting scratching to produce pain to help the itch in the same localization, if I've said that not too strangely. So they don't, if they scratch their hand, it doesn't help the itch of their face. And as to the types of pain, scratching is what they use. They sometimes use ice. And we have had one person in the big family I showed you who used dry ice. And of course, the problem with that was they then got burns from the dry ice. But they will go to quite extraordinary extent. They'll sit there with ice cubes on their face all day long if it's a hot summer. So
and
probably,
also, is this pain short term or long term that they want to keep it for, the pain?
oh, I think it's short term, because when you stop scratching it, normally allows them to reset themselves so that they can take a breather and stop scratching. They want to break the crescendo of the itch getting worse and worse. So sometimes just putting cold on their face will be enough. Other times they just can't help themselves, especially if they're a young child.
So it's not persistent pain that they need. They just need a break from the scratching to escape that.
Yes, that's what they say.
Thank you.
Thank you, Varghese. Professor, you have a comment in the comment box. Dr. Sarath Chandra. He says, very interesting presentation. He is a practicing dermatologist, clinical dermatologist. He also comments, now we can see our patients with idiopathic itch with a whole new approach. So there is a mess, yes.
Yeah, I think it's quite nice to have things to keep your brain active, isn't it, when you're in clinic and you're seeing what seems to be the same thing. It's quite nice to think there are other things out there. The other obvious hope is that, especially with the first family, maybe that's a new itch pathway which we can do something about. Maybe it's druggable. That would be the really exciting thing for this family, and of course probably for anyone, because itch pathways are like pain pathways. If you could block them, maybe you'd have something which would cure many different types of itches at one go. Well, I live in hope. I live in hope.
Professor, in terms of chronic disease, cancer, sickle cell, thalassemia, other diseases, we know there is pain, okay? And we also, I don't know, each related to pain, okay? That is one entity which you're talking now. And there are a lot of research going on in pain, in these diseases, especially cancer and sickle cell anemia, how to reduce the pain, And is there, do you think is a link between, they also must be having some itch? All pain should have itch? Or is it all itch should lead to pain? In terms of severity, you have very interestingly shown, one allele change,
So we think that the itch and the pain pathways overlap in some areas, especially in the spinal cord. So we think it's in the spinal cord where the itch sensation seems to be calculated, if that's the right word. And that's where pain can override it. Pain is a more important sensation to send up to the brain than itch. And so I think there are times they overlap. Opioids, opioids, one of the significant side effects is itch. Yeah, and we think we probably understand why that is, and maybe the first family are going to show us that. So it would be nice to have ways to stop patients having itch, because itch isn't always like I've showed. Itch can just be an irritation rather than a significant disease. If I may, I've got to go to another meeting now. So can I thank you very much and say good evening.
Thank you very much. Yeah.
Thank you.
Good night. Yeah.
Thank you.
Yes, Dr.
Yeah, thank you. Good job. And at least let us sum it up and then send a note for the benefit of all the people, both in the Times of India and Hindu for the readers across the country, and in Telugu, I think Inaudio and Jyoti. Let people know about this.
Yes.
Yeah, I think proper write-up and forwarding to Sastri may help other people to know about it.
Yeah. Dr. Gandhi,
yes.
This particular presentation,
Mm-hmm.
we have a group of 77 dermatologists.
I see.
It's in the dermatology group.
Mm-hmm.
Can you post this presentation? So
yes.
whoever could not be present today, they
yes.
can listen at least to his lecture.
Absolutely correct. So we can go ahead with that.
Yeah, Gargi, if you can give me that presentation, we can post, in
yes.
the group. I'll ask Ms. Gargi to look into.
Yes, she will do that. And from the public point of view, how to make it a readable version to be presented to the media. You try to work on that, and then for Telugu people, English people, for in English language, whatever you do it, Sastri can convert and send it to English media.
Okay.
Okay.
Okay. Thank you.
Thank you. I think good job and good person, good speaker.
Yeah.
I think both Anand Baggu and Sarachandra will spread this message of having interesting speakers for Genome Foundation webinars, so that others also will get benefited.
Right.
Okay then. Thank you.
Thank you.
Bye all. Bye all.
Bye. Thank you all. Yeah.
Thank you everyone for attending, and we'll keep in touch for the next series as well. I'll try to share the information as soon as possible.
Thank you, Vargi. Yeah.